Hello friends, welcome to today's session, Cell the Unit of Life. Myself Jalaja Sajeev, PGT Bio from KV Keshapuram. First we are going to an introductory session. Who discovered cell? You have covered this aspect even in your 9th and 8th class also. Robert Hooke in 1665 discovered a cell. Robert Hooke, an English botanist, the first cell discovered was bacteria, and it was discovered by Anton van Leeuwenhoek, who discovered protoplasm. Purkinje in 1839 discovered protoplasm, the physical basis of life. Now regarding the discovery of a cell, the basic unit of life, cell is the structural and functional unit of life of an organism. And as I have already told you, Robert Hooke in 1665 discovered a cell. Regarding his discovery, he took thin sections of cork. The scientific name of the plant is Corcus uber. Ultra thin sections were taken and observed under his rudimentary microscope. He found honeycomb-like structures, which he named it as cellulae, in Latin word which means compartments. Later they were named as cells. Why the cell is known as the functional unit of life? All the vital functions of life that can be carried out in a cell, and the cell contains different organelles for doing different functions. Thus, within a single cell itself there is division of labor. Each organelle is doing a specific function, vital function. For example, respiration is carried out by mitochondrion, digestion is carried out by lysosomes, and the reproduction is carried out by nucleus of the cell. Thus, within a single cell itself, even in the case of a unicellular organism also there is division of labor, but it is within the cell itself because of the different organelles. Now coming to the aspect of tissue, what constitutes a tissue? As we have already learned this concept in your earlier classes, a group of cells having same origin and a common function. A group of cells that have same origin and which will perform a common function, that constitutes a tissue. Study of cells, the branch of biology that deals with the study of cell. It is known as cytology. And the branch of biology that deals with the study of tissues, that is known as histology. Now regarding cell theory, M.J. Sheldon, a botanist, and T. Schwann, the zoologist, put forward a cell theory in 1838. It consists of the following facts: all the living organisms are made up of cells, and cell is the basic structural and functional unit of an organism. A cell originates from a pre-existing cell. The function of an organism as a whole is the sum total of all the functions of the constituent cells. Now, the cell theory that was modified by Rudolf Virchow, he stated that omnis cellulae cellula. It means a cell originates from a pre-existing cell. Now, coming to the advantages of multicellularity in an organism. The first point here comes division of labor. It helps in division of labor. Each cell is doing a specific function in order to avoid the duplication of labor. Death of a few cells do not kill the organism. For example, RBC cells as well as epithelial cells are replaced by new cells regularly, but the organism is not much affected. Some of the surface cells are dead, but they help in support as well as protection. Now, regarding the size and shape of the cells, it varies according to the functional status of a cell. They may be spherical, for example, RBC, then spindle shaped, for example, non-striated muscles as well as platelets, irregular in shape, amoeba and leukocytes, elongated in shape, nerve cells, neurons. So, thus the shape and size of the cell depends upon the functional status of the cell. Now, regarding the size also varies in various organisms depending upon its function. Bacterium, it is 3 mu m to 5 mu m, and in ostrich the egg cell, it is 170 into 135 mm in diameter, and mycoplasma the size varies from 0.1 mu m to 0.3 mu m. Now, coming to the differences between a prokaryotic cell and a eukaryotic cell. A very important question. We have to distinguish these two different types of cells based on some of the specific characters. Here you can see the differences. Prokaryotic cell comparatively smaller in size and eukaryotic cell is larger in size. Now, coming to the presence or absence of nucleus. In prokaryotic cell, nucleus is absent, well defined, and true nucleus is totally absent. In eukaryotic cell, nucleus is present. Now, coming to the organelles. Membrane bound organelles are totally absent in the case of a prokaryotic cell. It is present in membrane bound organelles are present in eukaryotic cells. For example, membrane bound organelles, mitochondrion, Golgi bodies, endoplasmic reticulum, lysosomes, all these comes under the category of membrane bound organelles. So, these membrane bound organelles are totally absent in prokaryotic cell and it is present in eukaryotic cell. Now, coming to the nuclear membrane. We have already seen nucleus, a well developed defined nucleus is totally absent. It means the nuclear membrane is also absent. but in eukaryotic cell you can see well developed nuclear membrane, a double layered nuclear membrane with the nuclear pores and all seen here. Now coming to the ribosome, the universal organelle present in this prokaryotic cell, it is the 70S ribosome, but in eukaryotic cell it is 70S and 80S ribosomes are present. Both the ribosome 70S and 80S are present. Now comes to this, at another important point is mesosomes present. Mesosomes are the infoldings of the plasma membrane that is present in the prokaryotic cell, and these mesosomes are present here in prokaryotic cell and it is absent in eukaryotic cell. Spindle fibers are not formed during cell division, and here well developed spindle fibers are formed during cell division. Now regarding the nucleolus, small spherical structure seen inside the nucleus, so that is totally absent here in prokaryotic cell, and in eukaryotic cell well developed nucleolus is also present. Now coming to another point, that is regarding the transcription and translation. Transcription, you know already, the copying of messenger RNA from one strand of the DNA, and translation is synthesis of protein from the copied mRNA based on the codons, sequence of codons and alignment of specific amino acids. So these two processes, transcription and translation, both will take place simultaneously in the cytoplasm. Since there is no developed membrane, nuclear membrane, this whole process that takes place in the cytoplasm. So this will take place simultaneously in the cytoplasm itself. But here in eukaryotic cell, first there occurs transcription. Then after that transcription, there will be the processing of mRNA, and after that processing the functional mRNA will be transferred to the cytoplasm, where there occurs the synthesis of protein, that means translation. So first there occurs transcription inside the nucleus, which is then followed by translation in the cytoplasm. Both can occur simultaneously. Now regarding the classification of bacteria based on shape, there are spherical bacteria, spherical in shape, we will call them it as coccus. Now there are different types of spherical bacteria. If it is seen singly, then we will call it as micrococcus, and if it is seen in a group of two, then we will call it as diplococcus. Diplo means two, and if it is seen in the form of a chain, then we will call it as streptococcus, and if it is seen in a group just like the bunch of grapes, then it is known as staphylococcus. So there are four different types of coccus, the spherical bacteria: micrococcus, diplococcus, streptococcus and staphylococcus. Example of a diplococcus bacteria, that is Diplococcus pneumoniae. Now if it is a rod shaped, then we will call it as bacillus. They may not have any flagella. They are of again different types. Streptobacillus, that means they are in the form of filaments or chain. For example is Bacillus subtilis. Vibrio, that is comma shaped bacteria. They are flagellated and capable of movement, so it is flagellated and motile. Example is Vibrio cholerae, causing the disease cholera. Now coming to the next one, that is spirillum, spring shaped, with a flagella at one end or both the ends. Example is spirillum. So, here you can see the different types of bacteria. Coccus type, this is Streptococcus. This one you can see here a group of 2, that is Diplococcus one. It's seen in groups, Staphylococcus, rod shaped bacteria, Bacillus, then spring shaped spirillum you can see here, and comma shaped one, that is Vibrio. So, these are the different types of bacteria based on their shape of the cell. Now, we can classify this bacteria based on staining also. The staining, that is gram staining. Based on gram staining, based on the color retained, we can divide the bacteria into 2 different groups. One is gram positive bacteria and the other is gram negative bacteria. Gram positive bacteria will retain the blue color when it is stained with a crystal violet dye, and when they are treated with, after treating with this gram stain, we will treat with the iodine as well as decolorized with alcohol, and even after decolorization with alcohol also it will retain the purple color, then that bacteria will be gram positive. And the gram negative bacteria, they will take the color of the counter stain, either it may be basic fission. So, that will be, the color will be pink in color rather than purple in color. So, pink colored bacteria, they are gram negative, and purple colored bacteria, they will be gram positive based on gram staining. Now, coming to the steps in gram staining. As I have already told you, here we are using the stain that is crystal violet. So, the primary stain, application of the primary stain, that is crystal violet, later we will be dealing here as CV, crystal violet. Heat and fix the smear. The bacterial smear that will be heated and fixed, after that it is stained with a crystal violet dye. The crystal violet dye will dissociate into Cv plus and Cl minus ions. These 2 ions then penetrate through the cell wall and cell membrane of both the gram positive as well as gram negative bacteria. Cv plus ions later interacts with the negatively charged bacterial components and stains the bacterial cells into purple in color. The second step is addition of grams iodine. Iodine is acting as a moderant in trapping the and act as a trapping agent. A moderant, that is a substance that will increases the affinity of a cell wall to a particular stain. It will bind to the primary stain, thus forming an insoluble complex which gets trapped in the cell wall. All the cells will then turn into purple in color. The third step here is decolorization with a 95% ethyl alcohol. Alcohol or acetone that will dissolve the lipid outer membrane of the gram negative bacteria. So, the peptidoglycan layer that get exposed and increases the porosity of the cell wall. The Cv iodine complex is then washed away from the thin peptidoglycan layer, leaving gram negative bacteria a colorless one. On the other hand, alcohol has a dehydrating effect on the cell walls of gram-positive bacteria. As a result, the pores of the cell wall shrinks. The CV iodine complex gets tightly bound into the multi-layered and highly cross-linked gram-positive cell wall. So, the cells get stained as purple in color. Now, the last step here is counter staining with safranin. The decolorized gram-negative cells are then counter stained with safranin, or basic fission can also be used instead of safranin, which will stain them pink in color. Now, coming to the structure of a bacterial cell or a prokaryotic cell in detail. The first part that we can see here is the cell wall, which is made up of murin or peptidoglycan. It is a polysaccharide cross-linked with short amino acid chains. Second layer, that is slimy gelatinous capsule. It is seen on the bacterial cell, which protects them from desiccation under the conditions of drought. The third layer, that is known as cell membrane, it will surrounds the protoplasm. Fourth one, the major part, that is the nucleus. A well organized and true well defined nucleus is totally absent in prokaryotic cell. No nuclear membrane and no nucleolus. So, we can call this particular structure as nucleoid. Oid means like, nucleoid means nucleus like structure, but not the true nucleus. It has a long DNA molecule which get attached with the mesosomes with the help of nucleoid proteins. Here you can see the structure of bacterial cell with the different layers, protective layers, the nucleoid structure, cytoplasm, pili or even sex pili, flagellum, all structures. Plasmid, an extracellular chromosomal, and circular DNA that is present in the cytoplasm of the cell. It can be used as a vector for transferring foreign DNA into the host organism in recombinant DNA technology. It contains antibiotic resistant genes that can be used as markers for identifying the recombinants from non-recombinants. Now, coming to cytoplasm, granular in nature and it is colorless, containing ribosome, the 70S ribosome, granules of a glycogen, fat and other food reserves. Other cell membrane bound organelles like mitochondrion, chloroplast, endoplasmic reticulum, Golgi bodies, etc. are totally absent in the prokaryotic cell. Pili, some of these bacteria contains small hair-like or rod-shaped structures called as pili, which help in the attraction of the opposite sex. So, it is known as sex pili. Flagellum originates from the basal granule seen in the cell membrane. In prokaryotic cells, the cell envelope contains 3 different layers, such as first one, that is glycocalyx. It is the outermost layer and it may be loose or it may be thick and tough layer. If it is a loose sheath, then it is known as slime layer. If it is thick and tough layer, then we will call it as a capsule. Both are made up of polysaccharides. Second layer, that is known as cell wall. Cell wall is the second layer of the cell envelope seen in bacteria or prokaryotic cell, which is seen just below the glycocalyx. It is rigid due to special macromolecule that is called as peptidoglycan. It is composed of a repeating framework of long glycan strands, that is N-acetyl muramic acid and N-acetyl glucosamine, which are cross-linked with short peptide chains. Plasma membrane made up of lipoproteins and oligosaccharides, not the steroids. Plasma membrane fold inwards in the form of vesicles, tubules and lamellae, which are called as mesosomes in prokaryotic cells. Now, coming to the functions of mesosomes, it increases the surface area of transport, helps in replication of DNA during cell division, helps in the formation of a cell wall during binary fission, helps in respiration, and it also helps in secretion. A prokaryotic cell contains many inclusions like phosphate granules, glycogen granules, cyanophycean granules. They contain gas vacuoles which help in positioning the prokaryotic cell in the water medium by giving buoyancy. Now coming to the structure of cell membrane. Cell membrane or plasma membrane or plasma lemma, the well accepted model suggested by S. Jonathan Singer and Garth Nicholson, that is the fluid mosaic model of plasma membrane. It is the most accepted model of plasma membrane. There are other models also, unit membrane model proposed by Robertson. So, here it is the most accepted one, that is the fluid mosaic model proposed by Jonathan Singer and Garth Nicholson. It is said that plasma membrane is formed of protein iceberg in a sea of lipids. Plasma membrane is trilaminar, it contains 3 layers and it is made up of lipoprotein. Lipid molecules are the phospholipids which form bilayer. Phospholipids itself will form 2 layers. Proteins are globular and are of 2 types, intrinsic and extrinsic proteins. Here you can see the diagram of fluid mosaic model. Phospholipid molecules are here, they are arranged in 2 different layers with the head region and tail regions. Then different other protein molecules, they are seen scattered in the lipid bilayer. Some other oligosaccharides, glycolipids, even cholesterol molecules are also here. Proteins which are seen completely inside the bilayer, lipid bilayer, that is known as intrinsic or integral protein. Proteins which are completely seen outside the lipid bilayer, they are known as extrinsic protein or peripheral protein. Some of the proteins, they are very large and project on both the surface of phospholipid bilayer, which are called as transmembrane proteins. They act as charged channels for the passage of water and soluble materials. Now coming to the structure of a phospholipid molecule. A phospholipid molecule is an amphipathic molecule which possesses a hydrophilic head and a hydrophobic tail. Head is made up of glycerol and phosphate, while tail is made up of fatty acid. Glycolipids or glycoproteins are the carbohydrates associated with the plasma membrane. Cholesterol molecules present in between the phospholipids, which provide stability to the animal membranes. Regarding phospholipids, more common types of phospholipids are phosphatidylcholine, known as lecithin, and the other one is cephalin, that is phosphatidyl ethanolamine, and yet another one, that is phosphatidylserine. So, these are the different types of phospholipids that are commonly seen in the plasma membrane. They allow the fast movement of fat soluble molecules. Cholesterol molecules stabilizes the plasma membrane. Plasma membrane helps in intracellular and extracellular transport of molecules. Now coming to cell wall, the outermost protective and rigid supportive layer that is seen in all the bacteria, cyanobacteria, protists and all the plant cells. It is absent in animal cell and it was discovered by Robert Hooke. Types of a cell wall, they present primary cell wall and secondary cell wall. Coming to primary cell wall, outer and thinner elastic and it is living, present in the meristematic cells and parenchyma cells. It is made up of cellulose, hemicellulose, pectin and protein. Second one is secondary cell wall. It is seen inner to the primary cell wall in a mature and non-dividing cells. It is made up of cellulose, hemicellulose and suberin in the case of cork cells, or cutin in all the aerial parts of the plants, or it may be lignin in the case of woody plants, and in grasses it is silica. Now regarding middle lamella, a thin and binding layer that is seen between the cell walls of adjacent plant cells. It is made up of calcium and magnesium pectates. Plasmodesmata, cytoplasmic connections between neighboring cells that takes place through the pits present in the cell wall. Now pits are the portions, unthickened areas present in the cell wall, and through these pits the cytoplasm of the neighboring cells can form connections, and that cytoplasmic connection, which is known as plasmodesmata, it helps in exchange of materials between the cells. an aggregation of a cytoplasm of neighboring cells that occurs through plasmodesmata. That unit is known as symplast. Now, components of the cell wall present in a plant cell: cellulose, hemicellulose, water molecules, pectic substances, proteins, and even lipids are present in the cell wall of plant cells. Now, coming to the functions of a cell wall, since it is rigid, hard, it gives protection. It gives a definite shape to the cell. It is involved in mechanical support. It helps in the absorption of water and mineral elements. For example, the cell walls of root hair absorbs water from the soil by imbibition. Suberin and cutin that will prevent evaporation of water or transpiration. Cytoplasm, an amorphous and translucent, homogeneous and colloidal ground substance that is seen between the plasma membrane and the nucleus. Cytoplasm can be differentiated into two parts. One is cytoplasmic matrix and the other is cytoplasmic structures. Cytoplasmic matrix is known as cytosol or hyaloplasm, which is again divided into ectoplasm and endoplasm. Cytoplasmic structures includes the different cell organelles, the living components and non-living components, that is cell inclusions. Now, regarding the different functions, one, it provides raw materials for cell organelles. It helps in exchange of materials. It helps in the biosynthesis of molecules. Now, regarding the vacuoles, it is present in all the eukaryotic cells. A single large vacuole that is seen in plant cell at the center, and in animal cells, small vacuoles are seen and they are scattered. A vacuole has two different parts. The outer membrane, just like that of plasma membrane, made up of lipoprotein in composition, that is known as tonoplast. The fluid content seen inside the vacuole, that is known as cell sap. Fluid content present inside the vacuole, and it contains different components like water, mineral elements, sugars, amino acids, proteins, alkaloids, and even pigments. Now, coming to the functions of vacuoles, cell sap exerts turgor pressure. Now, here comes the word turgor pressure, the pressure exerted by the expanding cytoplasm in a turgid cell against the cell wall. So, that pressure, that is known as turgor pressure. So, cell sap will exert turgor pressure and it helps in the cell growth, enlargement of the cell. It stores water and mineral elements as well as various types of nutrients. It stores even the waste materials of metabolism also. Now, coming to the different types of vacuoles, there are food vacuoles meant for the storage of food molecules. Contractile vacuoles are there, mainly they are seen in the protozoans. It is meant for osmoregulation as well as excretion. There are gas vacuoles, they help in floating or maintaining buoyancy. Now, here comes the organelle centriole. It was discovered by Van Benden It is seen in all the animal cells except in the case of mature mammalian RBCs. It is absent in plant cells. It is seen in protists as well as flagellated plant cells. It is totally absent in prokaryotes. Centrosome. It is the portion that is formed with the help of two darkly stained granules called as centrioles, and this, since they are two in number, they are known as diplosome. Here is the structure of a centriole with different filaments, microtubules. We will see the detailed structure here. Centrioles are surrounded by an amorphous and cytoplasmic area that is known as centrosphere. Centriole do not have any outer membrane in order to give a definite shape. So, it is amorphous and it is surrounded by a clear area that is known as centrosphere. Centrioles and centrosphere together we will call it as centrosome. Now, coming to the structure of a centriole, they are hollow cylinders. They are perpendicular to each other. They are two in numbers arranged perpendicular to each other, and each centriole is microtubular structure made up of microtubules. They are arranged in 9+0 manner. 9 peripheral tubules, and at the center there is no tubule at all. So, it is written as 9+0 configuration. Each microtubule is a triplet. There are 9 peripheral microtubule, and each microtubule is a triplet formed of 3 different subtubules that we can write or represent as A, B and C. A subtubule is formed of 13 parallel protofilaments. The single A microtubule of the peripheral tubule that is made up of 13 parallel protofilaments. B and C subtubules that is formed of 10 parallel protofilaments. So, there are total 9 peripheral tubules, microtubules, and each microtubule is a triplet represented as A, B, C, and A subtubule is made up of in turn protofilaments that is 13 in number, and B and C subtubules that is made up of 10 parallel protofilaments. C subtubule of each microtubule is linked to A subtubule of the adjacent microtubule by AC linker, and hence all the microtubules are a little bit tilted at 40 degree. At the center there is a proteinaceous hub. At the center of the centriole there won't be any microtubule at all. It is made up of a proteinaceous hub. The A microtubule that is connected with the central hub with the help of proteinaceous radial spokes. Now, coming to the functions of a centriole, they form asters during cell division in the case of an animal cell. They will form the basal body or blepharoplast of the flagella, which is seen in the plasma membrane. Distal centriole of the sperm that give rise to axoneme of the tail region of the sperm. Now, here are some of the questions for you to think and answer. Identify which among the following is not correct. Robert Brown discovered the cell. Schleiden and Schwann formulated the cell theory. Virchow explained that cells are formed from the pre-existing cells. A unicellular carries out its life activity within its cell. The second question for you to answer: new cells generate from A. Bacterial fermentation, B. Regeneration of old cells, C. Pre-existing cells, D. Abiotic materials. Think from the lessons that you observed. Now, here is your third question. Which of the following is correct? Cells of all living organisms have a nucleus. Both animal and plant cells have well defined cell wall. In prokaryotes, there are no membrane bound cell organelles. Cells are formed de novo from abiotic materials. So, out of these 4 options, which of the following is correct? That you have it to think and respond. Now, coming to the fourth question for you, it is a match the following question. Match the answers, questions and answers or options correctly. Cristae, cisternae, thylakoids. Here in the part B, options: flat membranous sacs in stroma, infoldings in mitochondria, disc shaped sacs in Golgi apparatus. Here are some more questions for you to answer. What is a mesosome? Mention the functions of it. The sixth question for you: how do neutral solutes move across plasma membrane? Can the polar molecules also move across it in the same way? If not, then how are these transported across the membrane? Think it properly and respond. Now, the next question for you: name 2 cell organelles that are double membrane bound. What are the characteristics of these 2 organelles? State their functions and draw the labeled diagram of both. So, all these questions you have to think it over and answer correctly. Thank you.